This lesson's about dihybrid crosses. We've already looked at Mendel's experiments where he crossed peas that differed at one trait controlled by one gene of interest. For example, crossing homozygous dominant yellow pea plants with homozygous recessive green pea plants. If this is the P generation, we followed through to the F1 and the F2 generation. Well, Mendel also did crosses where he looked at pea plants that differed at two traits controlled by two genes of interest. For example, in addition to color being controlled by the Y gene with yellow and green alleles, he also looked at the texture of the peas being controlled by another gene, which we'll call R. The dominant allele, capital R, codes for a round pea, and the lowercase r recessive allele calls for a wrinkled pea. And Mendel crossed plants that differed at both of these genes. For example, he would cross a plant that was homozygous dominant at both of them, that would give us a yellow round pea, with a plant that produced green wrinkled peas because it was homozygous recessive at both of these genes. The F1 generation from this cross, which is heterozygous at both loci, would be yellow and round because yellow and round are the dominant phenotypes. But then he would cross this again with another F1P, which is a second heterozygote. To think about the products of this cross, let's first start with the gametes that each of these plants could produce. They have the same genotype, they'd be able to produce the same gametes, so we'll just do it with one. For each of the Y and R genes, we have the option of the dominant or the recessive allele, and that leaves us with four possible combinations, ensuring that each gamete gets one copy of the Y and one copy of the R gene. We could have both of the dominants, we could have the two different combinations of one of the dominant alleles and one of the recessives, and we could have the two recessive alleles. So these are our possible gametes, and again that would be the same for both of these plants. So now we can put that into a Punnett square. I'll write them across the top here first, and they're going to be identical across the side because I'm crossing two plants from that F1 generation. Now we need a larger Punnett square here because we have more possible combinations, but we're going to approach it in exactly the same way. In each box we take the two gametes from the two different parents and we put them together to write the genotype that we're going to get. So here I have all dominant for both of the genes. Here I have two capital Y's and I have one capital R and one lowercase r. And I continue like that. So pause the video now and fill in the rest, and then you can check with what I filled in. So here are the genotypes all filled in. Now what about the phenotypes? If we first look for yellow and round, everything that has at least one capital Y and one capital R, we see them here. And if we circle them and then count them all up, we see that we have a total of 9. Now if we look for the yellow wrinkled, so capital Y but the recessive lowercase r and homozygous for the lowercase r, those are here. So these are going to be our yellow and wrinkled. If we look for green and round, this is our green and round here. These three. And green and wrinkled is just this one here in the corner. So counting those up, we see that we have a phenotypic ratio of 9 yellow round to 3 yellow wrinkled to 3 green round to 1 green and wrinkled. And this is a characteristic phenotypic ratio when you're crossing two organisms that are heterozygous at two different genes. And what's happening here is that we have two 3-to-1 ratios superimposed on top of each other. If you just look at color, then we've got a total of 12 yellow and a total of 4 green. Well, 12 to 4 is again a 3-to-1 ratio. And if you look at texture, we've got 9 round here and 3 round here, so that's a total of 12 round, and we've got 3 wrinkled and 1 wrinkled. Again, 12 to 4 is a 3-to-1 ratio. So we have two 3-to-1 ratios that are completely independent of each other, superimposed on top of each other, and that's why we get 9 to 3 to 3 to 1. And it's by looking at these two superimposed 3-to-1 ratios that Mendel derived his law of independent assortment, which states that the alleles of one gene separate into gametes completely independently from those of any of the other genes. In other words, the way we set up our Punnett square, where each of these 4 gametes was equally likely, was correct. We now know, of course, that independent assortment is due to the separation of homologous chromosomes in meiosis 1, because it's the homologous chromosomes that are carrying the alleles. But Mendel didn't know that. He just worked it out from his ratios. So that's the dihybrid cross.